Welcome to the Exam Podcast. Today we're not just dissecting a chapter, we're cracking the code of how you think, feel, and move. Imagine your body as the most complex supercomputer ever built. Today we're diving into its ultimate operating system, the neural system. How does it manage to orchestrate everything from a complex math problem to the simple act of breathing, all at lightning speed? That's our deep dive mission. Hello, tutor. What exactly are we exploring today in this essential deep dive into neural control and coordination?
Hello, and a warm welcome to all our dedicated learners. Indeed, today we embark on an essential deep dive into neural control and coordination. Our primary mission is, well, to unravel the incredible sophistication of our body's internal communication system. We'll start by understanding how our body maintains a stable internal environment, a critical process known as homeostasis, through precise and harmonious coordination among its many parts. We will then meticulously cover the intricate structure of the neural system itself, from its basic building blocks, the neurons, to its complex organization within us. What's truly fascinating and often a key area for competitive exams is the precise mechanism of nerve impulse generation and transmission. The electrical signals that power our thoughts and actions. Finally, we'll journey into the detailed architecture and functions of the human brain, the ultimate command and control center, exploring how this marvel is structured and what each part does. This knowledge is not just key to comprehending biology for your Class 11 board exams, but also forms the vital bedrock for competitive exams like NEET, AIMS, and UPSC General Science. It offers deep insights into real-world physiological phenomena you encounter every day.
Okay, that sounds like a grand tour. You mentioned coordination as the key to homeostasis, our body's internal balance. Can you elaborate on what coordination truly means in a biological context, tutor? Maybe beyond just things working together.
Certainly. Coordination in biology is far more than just things working together. It's a fundamental process where two or more organs interact and actually complement each other's functions. This ensures that all parts of the body work together efficiently and harmoniously towards, well, a common goal. Think of it as a perfectly synchronized orchestra, maybe, where each instrument plays its part not in isolation, but in response to and in harmony with others, all guided by a central score. This integration ensures our body responds effectively to both internal and external demands. Let's take a very relatable example to illustrate this. Physical exercise. When you start running or engaging in any strenuous activity, your muscles immediately demand significantly more energy.
Right. I definitely feel that.
So, to meet this heightened energy demand, your body needs a rapid and proportional increase in oxygen supply. This isn't a simple, isolated event. It necessitates a beautifully coordinated effort across multiple systems. Your respiration rate instantly goes up. You breathe faster and deeper, pulling in more oxygen. Simultaneously, your heart rate increases dramatically, pumping blood more rapidly to deliver that oxygen and nutrients to your working muscles.
Right, heart pounding, breathing hard.
Exactly. And blood flow through your vessels becomes faster, redirecting blood where it's most needed. Now, what happens when you stop exercising? All these systems, the nerves signaling the muscles, the lungs regulating oxygen intake, the heart pumping blood, even the kidneys regulating fluid balance, they all gradually and smoothly return to their normal resting states.
The cool down phase.
Precisely. This dynamic shift highlights the remarkable and precise coordination not just between muscles, lungs, heart, and blood vessels, but also involving the kidney, the liver, and many other organs. It ensures our body efficiently responds to changing demands and then recovers. It's a continuous dynamic balance.
That exercise example truly paints a vivid picture of how many systems have to work together. It's like a finely tuned orchestra indeed. But it also makes me wonder, tutor, if both the neural and endocrine systems handle this vital coordination, how do they, carve up the work? Do they ever step on each other's toes, or do they have distinct roles?
That's an excellent point, and understanding their distinct roles is fundamental for competitive exams. Our body primarily relies on two major systems for coordination and integration, the neural system and the endocrine system. While both are vital for maintaining homeostasis, their mechanisms and speed of action differ significantly. They're more complementary rather than competitive. The neural system provides a highly organized network of point-to-point connections. Imagine it like a dedicated, high-speed fiber optic internet connection, providing very quick, precise, and often localized coordination.
Super fast signals.
Exactly. It's composed of highly specialized cells called neurons that can detect, receive, and transmit different kinds of stimuli with remarkable speed and specificity. When you inadvertently touch something hot and instantly withdraw your hand, that's the neural system in action, rapid, direct, and localized. Its messages are electrical, traveling like lightning.
Okay, so neural is fast and direct. What about the endocrine system?
In contrast, the endocrine system provides chemical integration through hormones. It's more like a broadcast system, maybe, or a slower messaging service. Chemical messengers, hormones, are secreted directly into the bloodstream. These hormones then travel via the bloodstream throughout the body to target cells or organs that have specific receptors for them, often located far from the point of secretion.
So it's slower, but reaches more places.
Generally, yes. The effects of hormones are typically slower to initiate, but they can be more widespread and longer lasting. Think of processes like growth development or long-term metabolic regulation. These are primarily orchestrated by the endocrine system. So while both are vital, the neural system offers rapid, short-term, localized responses. The endocrine system provides slower, sustained, and often broader effects. This complementary action allows our body to fine-tune its responses to both immediate and long-term needs. It's a brilliant design for complex physiological control.
That makes sense. Fast action versus long-term regulation.
Precisely. Now, regarding the overall neural organization in animals, it's quite diverse and shows a clear evolutionary progression. It's fascinating, really. In lower invertebrates, like Hydra, for instance, the neural system is a very simple, diffuse network of neurons. Like a scattered web, no central processing unit.
Just a basic network.
Right. Then, as we move up to insects, the organization becomes more complex. They possess a rudimentary brain, several ganglia, which are like mini control centers, and various neural tissues. This allows for more complex behaviors.
A bit more organized.
Much more. And vertebrates, of course, represent the pinnacle. We possess the most highly developed, complex, and integrated neural systems with an elaborate brain and spinal cord, allowing for sophisticated behaviors, learning, and advanced cognitive functions like our own.
It's amazing seeing that complexity scale up. So, Tutor, now that we have this overview, let's focus specifically on our own human neural system. How is it fundamentally organized? What are its main components?
Okay, let's focus specifically on our human neural system. It's a remarkably organized network, but we can broadly divide it into two main parts for easier understanding. Think of it like a central processing unit and its extensive peripheral connections. First, we have the central neural system, CNS. This is the command center, the brain of the operation, quite literally. It's where decisions are made, information is processed.
The boss.
Pretty much. Second, we have the peripheral neural system, PNS. This acts as the extensive communication network, connecting the CNS to every other part of the body. Think of it as the vast cabling system allowing the central computer to interact with all its input and output devices.
That distinction makes perfect sense, like a central control tower and its fleet of aircraft. Tutor, could you elaborate on what exactly each of these parts, the CNS and PNS, includes? How do they function together?
Certainly. Let's break down each component's roles. The CNS is quite straightforward anatomically. It includes the brain and the spinal cord. That's it.
Brain and spinal cord. Got it.
It's the primary site for all information processing, decision-making, and control functions. All sensory info flows in, all motor commands flow out. It's the master integrator. The PNS, on the other hand, comprises all the nerves of the body that are associated with or extend from the CNS. These nerves are the communication lines, the wires. Now, PNS nerve fibers are further classified into two types based on the direction of impulse transmission. This is a key concept for exams.
Okay, direction matters. What are the types?
First, we have afferent fibers. These are essentially the sensory fibers. Think of them as reporters sending information to the CNS. They transmit impulses from tissues, organs, sensory receptors like skin, eyes, ears to the CNS.
So afferent brings information in, like feeling heat.
Exactly. When you touch something hot, the sensation travels via afferent fibers to your brain, always IN. Second, we have efferent fibers. These are the motor fibers. They are the command carriers. They transmit regulatory impulses from the CNS to the peripheral tissues or organs, initiating a response.
Efferent carries commands OUT, like pulling your hand away.
Precisely. After sensing heat via afferent fibers, the command to withdraw your hand comes via efferent fibers, always OUT. This rapid interplay between afferent and efferent pathways is fundamental.
Afferent in, efferent out. Simple enough, but crucial.
Very crucial. Now, the PNS itself is further divided into two major functional divisions based on conscious control. This distinction is vital. We have the somatic neural system. This relays impulses from the CNS specifically to our skeletal muscles. This system is largely involved in voluntary actions, movements we consciously decide to make.
Like picking up a pen, waving, things I choose to do.
Exactly. Lifting your arm, walking, typing notes, that's your somatic system sending signals to skeletal muscles. You're actively commanding it. In contrast, the autonomic neural system transmits impulses from the CNS to the involuntary organs and smooth muscles. These are actions we don't consciously control, but which are vital.
Like heartbeat, digestion, breathing while I'm focused.
Perfect examples. Your heartbeat, digestion, bladder contractions, even breathing when you're not thinking about it. These functions operate automatically, regulated by the autonomic system, maintaining our internal environment without conscious effort.
That's a very clear distinction between voluntary and involuntary actions. Tutor, could you give us maybe another concrete, everyday example to differentiate these two? Something that really highlights how automatic things are.
Absolutely. Let's use an example that might even be happening right now as you listen. A voluntary action, like consciously deciding to shift your posture in your chair or maybe scratch an itch, is governed by your somatic neural system.
Okay, I just did that.
See, your brain, part of the CNS, decided to move, sent signals via efferent somatic fibers to the specific skeletal muscles needed, and you moved. You controlled it. Conversely, an involuntary action, like the slight adjustments your blood vessels are making right now to maintain blood pressure, or perhaps the secretion of saliva in your mouth, is entirely under the control of your autonomic neural system.
Didn't even notice those things happening.
Exactly. You aren't consciously telling your blood vessels to constrict or dilate, or your salivary glands to produce saliva. These happen automatically, regulated by autonomic fibers. They just happen, continuously, maintaining your life without your conscious effort. Delving deeper into the autonomic neural system, it is in turn further classified into two subdivisions. These often work in opposition, creating a dynamic balance, very important for exams.
Opposing forces, like acceleration and brakes.
That's a great analogy. First, the sympathetic neural system. This is the accelerator associated with fight or flight responses. It prepares the body for stressful or emergency situations. When activated, it increases heart rate, breathing, dilates pupils, inhibits digestion, diverts blood to muscles.
The adrenaline rush, like when you nearly trip.
Exactly that feeling. That's your sympathetic system kicking in. Danger, react. Second, the parasympathetic neural system. This is the brake associated with rest and digest functions. Its role is generally the opposite, to conserve energy during calm periods. It slows heart rate, constricts pupils, stimulates digestion, promoting relaxation and recovery.
So after the near trip, when you calm down, that's parasympathetic.
Precisely. It says, okay, calm down, conserve energy, digest lunch. These two systems often have antagonistic effects on the same organs, maintaining a dynamic balance or equilibrium. Finally, we have the visceral nervous system. This is actually considered a part of the peripheral nervous system. It's a complex network of nerves, fibers, ganglia, and plexuses, specifically carrying impulses between the CNS and the viscera, our internal organs. It's crucial for regulating functions of the stomach, intestines, heart, lungs, often working alongside the autonomic system.
Okay, that's an incredibly detailed breakdown of the organization, from the big picture down to automatic control. So we've covered the systems and pathways. Now, Tutor, I imagine the real heroes are the individual cells doing the transmitting. Let's zoom in. What about the fundamental building block? The neuron. What makes it so special?
Indeed, you're absolutely right. Now let's zoom right in on the fundamental unit, the neuron, or nerve cell. A neuron is microscopic but incredibly complex and efficient. It's the structural and functional unit of the neural system. It's designed with remarkable precision to detect, receive, and transmit information. And what makes it special is that it's excitable. It can generate and transmit electrical signals. Let's take this step by step and visualize a typical neuron, perhaps like the one in your NCERT textbook diagrams. Understanding its structure is key. A neuron is primarily composed of three major parts. First, the command center. The cell body, also called the soma. This contains the nucleus, cytoplasm, typical organelles like mitochondria. But what's unique and characteristic here are the granular bodies called Nissl's granules. Remember that name?
Nissl's granules. Okay, noted. What's next?
Then, radiating out like branches from the cell body, we have the dendrites. These are short, repeatedly branching fibers. Importantly, they also contain Nissl's granules. Their main role is to transmit nerve impulses towards the cell body. Think of them as the antenna receiving signals from other neurons or receptors.
Receiving signals. Got it. And the third part?
And finally, extending out, usually much longer, we have the axon. This is typically a single long fiber. Its far end, the distal end, is usually branched, forming terminal arborizations. Each branch ends in a bulb-like structure called a synaptic knob.
Synaptic knob. Sounds important.
Very important. These knobs contain tiny sacs called synaptic vesicles, filled with chemical messengers called neurotransmitters. We'll come back to those. The axon's job is to transmit nerve impulses away from the cell body, sending the signal onward to another neuron or a muscle or gland.
Okay, cell body, dendrites receive, axon sends away via synaptic knobs. That makes sense. And those Nissel's granules you mentioned in both the cell body and dendrites, what is their specific function? Are they just there?
Good question. No, they're functionally very important. Nissel's granules are essentially clumps of rough endoplasmic reticulum and free ribosomes.
Ribosomes, so protein synthesis.
Exactly. Their main function is protein synthesis. This tells us neurons are metabolically very active. They constantly need to produce proteins for growth, repair, maintaining structure, and importantly, for producing neurotransmitters and other molecules needed for signaling. They're the neuron's protein factories.
So vital for keeping the neuron running.
Absolutely. Now, neurons can also be classified based on their structure, specifically the number of axons and dendrites. Multipolar neurons, these are the most common. One axon, two or more dendrites. Found abundantly in the cerebral cortex for integrating lots of information.
One axon, many dendrites. Common in the brain.
Bipolar neurons, less common. One axon and one dendrite. Classic example in the retina of your eye. For vision, a more direct relay.
One axon, one dendrite, like in the eye.
Unipolar neurons. These have a cell body with just one process extending out, which then splits. Found mainly during embryonic stages, but also in some sensory ganglia in adults.
Okay, different structures for different roles.
Precisely. Furthermore, axons themselves can be of two types based on their covering, and this difference dramatically affects signal speed. This is critical. We have myelinated nerve fibers. These axons are wrapped by specialized glial cells called Schwann cells, which form a fatty insulating layer called the myelin sheath.
Myelin sheath, like insulation on an electrical wire.
Exactly like that. And this sheath isn't continuous. There are gaps, periodic interruptions called the nodes of Ranvier. This myelination massively increases the speed of nerve impulse conduction. The impulse essentially jumps from one node to the next.
Jumps? How does that work?
It's called saltatory conduction, from the Latin saltare, to jump. It's much faster than continuous conduction. You find these myelinated fibers in spinal and cranial nerves where speed is essential.
Okay, myelinated means fast, jumping conduction via nodes of Ranvier. What's the other type?
The other type is non-myelinated nerve fibers. These are also enclosed by Schwann cells, but the cells don't form that thick myelin sheath. Impulse conduction here is slower and continuous, traveling along the entire membrane surface. You'll find these in the autonomous and somatic systems, perhaps where maximum speed isn't quite as critical. Understanding myelination and saltatory conduction is absolutely key for exams.
Here's where it's really interesting, Tutor. So these neurons are excitable cells. They generate and transmit electrical signals. But how do they actually do that? What's the secret behind them being excitable, and how does that signal zip along an axon?
Yes, this is the core mechanism. It's truly where the magic of neural communication happens. Neurons are unique because their membranes can rapidly change their electrical potential. This excitability comes from their membranes being in a precise, polarized state when at rest. Think of it like a tiny charged battery ready to fire. Understanding this step by step is crucial.
Okay, break it down for us. Resting state first.
Right. Let's start with the resting potential, the polarized state. When a neuron is not conducting an impulse, its membrane maintains an electrical difference across it. The membrane has various ion channels, like selective gates. At rest, the axonal membrane is comparatively more permeable to potassium ions, K+, but nearly impermeable to sodium ions, Na+. Also importantly, it's impermeable to large, negatively charged proteins trapped inside the axoplasm, the axon cytoplasm.
So more K+ can leak out than Na+ can leak in. And big negative proteins are stuck inside.
Essentially, yes. Consequently, inside the axon, you have a high concentration of K+ in these negative proteins, but low Na plus S. Outside the axon, it's the opposite. Low K plus S, high Na plus S. This creates ionic concentration gradients. These gradients are actively maintained by the sodium-potassium pump. This pump uses energy, ATP, to actively transport 3 Na plus ions outwards for every 2 K plus ions it transports inwards.
Pumping more positive charge out than in?
Exactly. That contributes significantly. So, due to differential permeability and this active pump, the outer surface of the membrane gets a net positive charge, while the inner surface becomes negatively charged. This electrical potential difference across the resting membrane is the resting potential, typically around negative 70 millivolts. The membrane is polarized, charged, and ready.
Okay, resting potential is negative 70 milliVs, polarized, more No plus outside, more K plus inside, ready to go. Now, what happens when it gets a signal?
Now for the action. Part 2. Generation of an action potential, or depolarization. When a stimulus of sufficient strength, a threshold stimulus, hits the neuron, it triggers a dramatic change at that site. The voltage-gated sodium channels at that spot suddenly swing open. The membrane becomes freely permeable to NAP plus ions.
NAP plus gates open.
Yes, and because there's so much NAP plus outside and it's positive, it rushes into the axoplasm down both its concentration and electrical gradients. A rapid influx. This flood of positive NAP plus ions reverses the polarity at that site. The outer surface becomes negative and the inner side becomes positive.
The inside becomes positive. Flipped.
Completely flipped, momentarily. The membrane at that site is now depolarized. This rapid, transient reversal of potential is the action potential. That is the nerve impulse.
Wow. So the impulse is that flip in polarity.
Yeah.
How does it travel then?
Good question. Part 3. Conduction of the nerve impulse. The action potential at one spot creates local currents that trigger the same thing in the adjacent spot. It propagates like a wave or dominoes falling. At the site immediately ahead, the membrane is still polarized, outside positive, inside negative. A local current flows on the inner surface from the now positive, depolarized site A, to the still negative polarized site B ahead. Simultaneously, on the outer surface, current flows from the positive site B back to the negative site A, completing the circuit.
Like a little electrical circuit flowing forward.
Exactly. This local current flow is the trigger for the next section. It causes the voltage-gated Na plus channels at site B to open. Na plus rushes in, site B depolarizes, generates its own action potential. And this sequence repeats rapidly along the entire length of the axon. That's how the impulse is conducted.
So it regenerates itself all the way down. Amazing. What happens after it passes? How does it reset?
Excellent point.
Yeah.
The final part, repolarization. The neuron needs to reset quickly. The massive increase in Na plus permeability is extremely short-lived. Those channels slam shut very quickly. Almost immediately after voltage-gated potassium channels open, now the membrane becomes very permeable to K plus Ren.
K plus gates open now.
Yes, and since there's much more K plus inside, and the inside is now positive, K plus ions rush out of the cell down their gradients. This outflow of positive K plus ions restores the negative charge inside and the positive charge outside. It brings the membrane potential back towards the resting potential.
So K plus rushing out resets the charge.
Correct. This is repolarization. The membrane quickly returns to its polarized state, ready for the next impulse. The sodium-potassium pump then works to fully restore the original ion concentrations over time.
That step-by-step breakdown makes it much clearer. It's a carefully orchestrated dance of ions, an electrical wave. So once an impulse travels down one neuron, how does it bridge the gap to the next neuron or to a muscle cell? You can't just jump electrically most of the time, right?
Exactly. You've hit upon the next critical step. Transmission across the synapse. A nerve impulse needs to be transmitted from one neuron to the next, or to an effector cell like a muscle or gland. This occurs at these specialized junctions called synapses. A synapse is basically the junction formed by the membranes of a presynaptic neuron, the one sending the signal, and a postsynaptic neuron, the one receiving it. These membranes might be extremely close or separated by a tiny cap called the synaptic cleft.
Okay, the junction point. Are there different types of synapses?
Yes, there are two primary types. First, electrical synapses. Here, the membranes are in very close proximity, connected by channels called gap junctions. Electrical current, ions, can flow directly from one neuron to the other. It's like the impulse continues almost uninterrupted.
It's a super fast transmission.
Very fast, yes. Faster than chemical synapses. But they are quite rare in our system, found where extreme speed and synchronization are needed. Second, and much more common, are chemical synapses. Here, the pre- and postsynaptic membranes are separated by that fluid-filled space, the synaptic cleft. There's a physical gap.
A gap. How does the signal cross the gap then?
Through chemicals, specifically chemicals called neurotransmitters. They are essential messengers at these synapses. Let's break down the mechanism at a chemical synapse step by step. It's a key process. First, remember those axon terminals, the synaptic knobs on the presynaptic neuron. They contain numerous synaptic vesicles filled with neurotransmitters like acetylcholine, dopamine, serotonin, etc.
The chemical messengers are stored there, ready to go.
Exactly. Now, second part. When an action potential arrives at the axon terminal, it triggers voltage-gated calcium channels to open on the presynaptic membrane.
Calcium channels now, not sodium or potassium.
Right. Calcium, CO2+, rushes into the terminal. This influx of calcium is the crucial trigger for the next step. Third part. The calcium influx stimulates the synaptic vesicles to move towards the presynaptic membrane. Fourth part. The vesicles fuse with the membrane and release their neurotransmitters into the synaptic cleft. This release process is called exocytosis.
They dump the neurotransmitters into the gap.
Precisely. Fifth part. The released neurotransmitters diffuse across the narrow synaptic cleft. They then bind to specific receptor proteins located on the postsynaptic membrane. It's a highly specific binding, like a key fitting into a lock.
Lock and key. Okay.
Yeah.
What happens when they bind?
Finally, the sixth part. This binding opens ion channels on the postsynaptic membrane. This allows ions to flow into or out of the postsynaptic neuron, generating a new electrical potential there. This new potential might be excitatory, making the postsynaptic neuron more likely to fire.
Like hitting the gas pedal.
Good analogy. Or it might be inhibitory, making it less likely to fire.
Hitting the brakes.
Exactly. It depends entirely on the specific neurotransmitter and the specific receptor it binds to. This balance of excitation and inhibition is crucial for complex information processing.
That's a very clear explanation of such a complex process, Tutor. I'm curious, can you elaborate a bit more on what specifically makes a potential excitatory or inhibitory? How does binding lead to go or no-go?
Certainly. It comes down to which ion channels are opened by the neurotransmitter receptor interaction. When an excitatory neurotransmitter binds, it typically opens channels that allow positive ions, most commonly sodium ions, Na+, to enter the postsynaptic neuron.
Na+ rushing in again.
Yeah.
Like in the action potential.
Similar effect, yes. This influx of positive charge causes a partial depolarization of the postsynaptic membrane called an excitatory postsynaptic potential, or EPSP. It makes the inside less negative, pushing it closer to the firing threshold. If enough EPSPs sum up, they can reach the threshold and trigger a full action potential in the postsynaptic neuron. It increases the chance of firing.
Okay, excitatory Na plus N equals closer to firing. What about inhibitory?
An inhibitory neurotransmitter works to prevent firing. When it binds, it might open channels for negative ions like chloride ions, Cl, Cella, to enter.
Negative ions coming in.
Yes, making the inside even more negative. Or it might open channels allowing positive potassium ions, K+, to leave the cell, also making the inside more negative. This increase in negativity is called hyperpolarization, or an inhibitory postsynaptic potential, IPSP.
Hyperpolarization.
Yeah.
Making it harder to fire.
Exactly. It moves the membrane potential further away from the threshold, making it much harder for excitatory signals to trigger an action potential. It acts as a brake. This constant interplay, this summation of EPSPs and IPSPs arriving from possibly thousands of other neurons, determines whether a given neuron will fire its own action potential. It allows for incredible nuance in processing.
Wow, it's like a constant calculation happening at every synapse.
Yeah.
So after these impulses are generated and transmitted, where does all this information ultimately go? Moving on to the pinnacle of the brain, Tutor, how does this command and control system integrate everything? What makes it so powerful?
The brain. Yes, the central information processing organ. It truly acts as the command and control system. Its capabilities are, well, astonishing. It's responsible for an incredible array of functions, controlling all voluntary movements, maintaining balance and posture, regulating vital and voluntary organs, lungs, heart, kidneys, handling thermoregulation, controlling hunger and thirst, managing our circadian rhythms, our 24-hour clocks. It oversees endocrine glands, linking nervous and endocrine control. And fundamentally, it governs human behavior.
That's a huge list already.
And there's more. The brain is the site for processing all our senses: vision, hearing, speech, smell, taste, touch, giving meaning to the world. It's the seat of complex cognitive functions: memory, intelligence, emotions, thoughts, learning, reasoning, creativity, feelings. It all happens here. It's what makes us human.
Truly the most complex thing we know. Given its importance, how is it protected?
Exceptionally well protected, as it must be. Firstly, it's encased within the sturdy skull, solid bone. Secondly, inside the skull, it's covered by 3 protective membranes, the cranial meninges. You need to know these layers from outside to inside. Outermost is the duramater, tough, fibrous, like heavy-duty armor.
Duramater, tough outer layer.
Middle layer is the arachnoid, thin, web-like, delicate.
Arachnoid web-like middle.
Innermost is the pia mater, very delicate, clings directly to the brain surface, highly vascular, the tender mother.
Pia mater, delicate inner layer touching the brain. Skull, then dura, arachnoid, pia. Got it.
Excellent. Now, the brain itself, structurally, is broadly divided into 3 major parts. You can see this in diagrams like a sagittal view. These are 1, forebrain and midbrain, and 3, hindbrain.
Forebrain, midbrain, hindbrain. Okay, let's explore each of these major divisions. Tutor, what can you tell us first about the forebrain? Sounds like the most complex part.
It is indeed the largest and most complex part, responsible for our higher cognitive functions. The forebrain consists of 3 primary components: the cerebrum, thalamus, and hypothalamus. The cerebrum is the major dominant part. It's divided longitudinally into two halves, the left and right cerebral hemispheres.
The two big halves we usually picture as the brain.
Exactly.
Yeah.
These hemispheres are connected by a massive tract of nerve fibers called the corpus callosum, allowing communication between them. The outer layer covering the hemispheres is the cerebral cortex. It's highly folded, with ridges, gyri, and grooves, sulci.
The wrinkles, why are they there?
Those folds dramatically increase the surface area, allowing for many, many more neurons to be packed in, more processing power. The cortex is referred to as gray matter because it's rich in neuron cell bodies, dendrites, and unmyelinated axons, giving it a grayish look. This cortex contains specialized regions: motor areas for voluntary movement control, sensory areas for interpreting sensory info, and large regions called association areas. These are neither purely sensory nor motor.
Association areas, what do they do?
They're crucial for complex functions like integrating different senses, memory formation and retrieval, language, reasoning, consciousness. Basically, our higher-level thinking. Deep to the gray matter cortex is the white matter. This consists mainly of myelinated nerve fibers, giving it a white appearance. It's the communication wiring within the cerebrum and to other brain parts.
Gray matter processes, white matter connects. Makes sense. What about thalamus and hypothalamus?
Right. The cerebrum wraps around a key structure called the thalamus. Think of it as a major relay station. Almost all sensory information, except smell, passes through the thalamus before going to the correct part of the cortex. It also plays roles in motor control, sleep, and alertness.
The brain's switchboard.
A very good way to put it. Then below the thalamus is the hypothalamus. Small but incredibly important, it controls vital functions: body temperature, hunger, thirst, water balance. It also contains neurosecretory cells producing hormones that regulate the pituitary gland.
So it links the nervous and endocrine systems directly.
Yes, a critical link. Finally, within the forebrain, inner parts of the hemispheres and associated deep structures like the amygdala, emotion, and hippocampus memory form the limbic system. Along with the hypothalamus, the limbic system is involved in regulating emotions: pleasure, rage, fear, motivation, sexual behavior, and memory, our basic drives and feelings.
Wow, the forebrain handles a lot. What about the midbrain?
The midbrain is smaller, located between the forebrain's thalamus hypothalamus and the hindbrain's pons, a vital bridge. A canal called the cerebral aqueduct runs through it, connecting fluid-filled ventricles. Its dorsal part has 4 round swellings called corpora quadrigemina. These are involved in visual and auditory reflexes, like automatically turning your head towards a loud noise.
Reflex centers, got it. And the hindbrain?
The hindbrain comprises 3 essential structures: the pons, cerebellum, and medulla oblongata, crucial for basic functions and coordination. The pons lies just above the medulla. It consists mainly of fiber tracts connecting different brain regions, especially the cerebrum and cerebellum. Also helps regulate breathing.
Pons connects things. What about the cerebellum? It looks distinct.
The cerebellum is very distinct, highly folded like a mini cerebrum. That folding allows for a huge number of neurons. It's absolutely crucial for coordinating voluntary movements, making them smooth, precise, and balanced. Also vital for posture, balance, and learning motor skills like riding a bike.
The coordination center for movement.
Exactly. It integrates sensory information from the body and inner ear to fine-tune motor commands. Finally, the medulla oblongata, or just medulla, it connects to the spinal cord. This is a critical control center. It contains centers regulating vital involuntary functions: respiration, heart rate, blood pressure, swallowing, vomiting, basic life support.
Medulla controls essential life functions. Damage there would be very bad.
Extremely dangerous, yes. Now, together, the midbrain, pons, and medulla form the brainstem. The brainstem connects the cerebrum and cerebellum to the spinal cord. It's the main pathway for information flow and houses many vital reflex centers.
Tutors, that's incredibly comprehensive. The cerebrum and cerebellum both seem involved in movement. Can we quickly compare their functions again? That seems like a common point of confusion.
Certainly an excellent point to clarify for exams. Think of it simply like this. The cerebrum initiates the voluntary movement. It's the part that decides, I want to pick up that pen. It handles the planning and conscious command.
Cerebrum decides and starts the action.
Yes. The cerebellum then acts like a coordinator or editor. It doesn't start the movement, but it refines it as it happens. It receives sensory feedback about your body position and the movement itself, and constantly adjusts the signals to make the movement smooth, accurate, and coordinated. It ensures you don't overshoot, undershoot, or tremble. It also maintains your balance and posture during the movement.
So cerebellum fine-tunes and coordinates what the cerebrum started.
Precisely. Cerebrum is the planner and initiator. Cerebellum is the coordinator and refiner. That distinction is key.
That analogy perfectly clarifies their distinct roles.
Yeah.
Thank you, tutor.
You're welcome. So in summary today, we've taken a really deep dive into neural control and coordination. We started with coordination itself, contrasted the rapid point-to-point neural system with the slower chemical endocrine system. We detailed the CNS, brain, spinal cord, and PNS, breaking down the PNS into afferent efferent fibers and the somatic autonomic systems. We explored the sympathetic and parasympathetic divisions and the visceral system.
Cover the whole organization.
Then we focus on the neuron cell body, dendrites, axon, Nissel's granules, myelination, nodes of Ranvier, saltatory conduction. Critically, we broke down nerve impulse generation, resting potential, polarized, action potential, depolarization via naught plus influx, conduction and repolarization via K plus efflux.
The whole electrical signal process.
Exactly. Then synaptic transmission, electrical versus chemical synapses, the role of neurotransmitters, calcium influx, vesicle fusion, receptor binding, and excitatory EPSP versus inhibitory IPSP potentials.
Bridging the gap between neurons.
Right. And finally, the brain. Its protection, skull, meninges, the major divisions, forebrain, midbrain, hindbrain, and the key structures within each. Cerebrum, cortex, gray-white matter association areas, thalamus, hypothalamus, limbic system, corpora quadrigemina, pons, cerebellum, medulla, and the brainstem.
A complete tour of the command center.
Indeed, this chapter is truly foundational. My advice, revise this topic again, paying close attention to the diagrams, the neuron, the brain in your NCERT book. Visualize them. Practice drawing and labeling them. Work through previous exam questions, especially on impulse conduction and synaptic transmission. Try to understand the why behind each step, not just memorize facts. That really helps with retention and problem solving. And of course, feel free to reach out if any doubts remain about this chapter. Keep learning, keep practicing, and keep excelling.